Now let's come to the different types of asexual reproduction. The first type, as you can see, is binary fission, which means the parental organism here divides into two halves. Each half receives equal genetic material and is able to grow into a new individual. On the basis of how they divide into two halves, binary fission is again of three types: simple binary fission, transverse binary fission, and longitudinal binary fission. In simple binary fission, the plane of division occurs through any direction. There is no particular direction of division. From any direction, the organism divides into two halves. Example, amoeba. In transverse binary fission, the organism divides along the transverse axis of the animal into two halves. Transversely, it divides. And for this, we can see in paramecium. And longitudinal binary fission, we find the division occurring along the longitudinal axis of the animal body, dividing the body into two halves. Example in Euglena. Next, we come to budding. In budding, a small projection is formed from the parental animal, and each the projection is known as bud. Each bud then separates from the original animal, from the parental body, and now starts growing into a new individual. We find budding in yeast, in hydra, most commonly. Next, fragmentation. In fragmentation, the body of the parent is broken down into many small parts called fragments, and each fragment under suitable condition gives rise to new individuals by regeneration. We find it in planaria. Even in hydra, we find fragmentation also. Next, sporulation. During adverse conditions, amoeba withdraws its pseudopodia and it secretes a cyst around itself. And when the conditions of the atmosphere again return back to normal, the cyst bursts, and from the cyst comes out several small amoeba which have been formed by multiple fission within the cyst. Next, we come to gamule formation. In some sponges, we find internal buds developing inside the parental body. These internal buds are called gamule, and each gamule develops into new individual. And lastly, we come to spore formation. In organisms like fungi mainly, special structures are present for asexual reproduction. Some are called zoospores, which are motile spores, which can move about. Example, Chlamydomonas. Sporangiospore, we find it in Rhizopus, or what you know as common bread mold. Sporangiospores are spores, but they are non-motile. And in Penicillium, we find Conidia. These are also non-motile spores, but they do not develop inside the sporangium. They develop freely on the surface. We find it in Penicillium. Now we come to vegetative reproduction in plants. Vegetative reproduction means development of new plants from the vegetative part of the parental plant, like stems, from roots, from leaves. So these are the vegetative parts, not the reproductive parts. From the vegetative parts, new individuals develop. For example, we have tuber, modified underground stem. We find tuber in potato. Rhizome, another modified underground stem. We find it in ginger, in turmeric. Next, we have bulbil. Bulbil is actually specialized, fleshy and rounded bud-like structures. These structures can give rise to new plants after detachment from the parent plant. We find it in agave, pineapple, oxalis. Next, bulb. Bulb is again another modified underground stem found in onion, garlic. Then we have offset runner sucker. These are actually creeping stems. They run horizontally on the soil surface. Runners are found in grass. Stolons are also found in Vallisneria, whereas stolons are not in the scope, but stolon is also one of them. Runners we find in grass. Offset, you must have seen offset around Dankuni very frequently. Water hyacinth, and suckers found in pineapple.